Muchas gracias. Bienvenidos a este evento que es muy importante para nosotros porque estamos celebrando los 50 años de fundación de nuestro Departamento de Biología Celular. Entonces tengo el privilegio y el honor de presentar al doctor Francisco Javier Quintana. Él es profesor de Neurología en el Romney Center de Neurologic Diseases at Brigham en Women's Hospital en Harvard Medical School y es miembro asociado del Broad Institute en Harvard y del MIT, del Massachusetts Institute of Technology. El doctor Quintana también es presidente de la International Society of Neuroimmunology. Él obtuvo su grado por la Universidad de Buenos Aires en 1999 y hizo un doctorado en inmunología en el Instituto Weizmann en Israel. Él realizó entrenamiento postdoctoral también allí en el Weizmann Institute of Science y posteriormente en la Harvard Medical School. En el 2009 se unió a la facultad de la Harvard Medical School. Y el doctor Quintana pues, ha estado desarrollando varios temas que están enfocados en neuroinmunología, donde investiga rutas de señalización que controlan la inflamación y la neurodegeneración, con el fin último de encontrar blancos terapéuticos para enfermedades neurodegenerativas y mediadas por el sistema inmune. El doctor Quintana ha publicado más de 230 artículos científicos y capítulos de libros, y uno de sus hallazgos más importantes pues, fue la identificación del gen que codifica el receptor para los hidrocarburos de arilo, que es un importante regulador en la inflamación de las células, tanto adaptativas como del sistema inmune innato. Y él ha definido mecanismos a través de interacciones célula-célula, microbiomas, metabolismo y de componentes químicos ambientales que controlan la inflamación y la neurodegeneración. Además, el trabajo del doctor Quintana pues, condujo a un medicamento que se llama Tapinarof, que fue aprobado por la FDA para tener como blanco terapéutico precisamente este receptor, el AHR, para el tratamiento de la psoriasis. El doctor Quintana pues, ha recibido múltiples premios, destacándose el Lady Anne Chain Prize for Academic Excellence and Scientific Achievements, the Junior Investigator Award from the National Multiple Sclerosis Society, the Pathway of Independence Awards of the National Institute of Allergy and Infectious Diseases, the Award for Outstanding Research Achievement from Nature Biotechnology, the Tican Award for Innovation, the Harry Weaver Award from the National Multiple Sclerosis Society, the Mentor Award from Harvard Medical Schools, the Milestones in Multiple Sclerosis Research Award from the National Multiple Sclerosis Society, the American Association of Immunologists BD Biosciences Investigator Award, most highly cited list, the Baron C. Prize of Innovation Multiple Sclerosis Research, el Ignite BWH Award, and the Rices Prize for Excellence in Research, esto en Argentina. Y en el 2021 el doctor Quintana fue nombrado el Kuchroo Weiner Distinguished Professor of Neuroimmunology. Y el doctor Francisco Quintana, como bien saben, va a dar dos charlas. La primera, el día de hoy, es sobre el control de la inflamación del sistema nervioso central a través de interacciones célula-célula. Entonces, brindémosle un fuerte y un caluroso recibimiento al doctor Quintana. Y adelante, profesor.
Bueno, antes que nada, mientras vienen los slides, bueno, primero muchas gracias por la invitación, es un placer realmente estar acá, conocer gente, conocer el instituto, la primera vez que he conocido el DF, que también desde el punto de vista personal es muy interesante. Muchas gracias por la presentación, realmente. Una nota es que yo no cloné el receptor de IHR y Guillermo Elizondo y otros pueden explicar más la historia de eso, pero trabajo mucho en ese receptor. Y por último, lo que quiero avisar es que por el bienestar de ustedes, para que no tengan que lidiar ni con mi acento argentino y con el 50% de las palabras en inglés, voy a tratar de dar mi charla en inglés. Me parece que va a ser lo mejor para todos. Bueno, y con eso, so with that in mind, what I want to do is, I'm very interested in what controls CNS inflammation, CNS, the brain, and in particular, when we think about inflammation in the central nervous system, we are mostly focused in a disease which is called multiple sclerosis. Multiple sclerosis is an autoimmune disease by which the immune system, and we'll go in detail over its mechanisms of pathogenesis in a sec. But basically what you have is an autoimmune response. The immune system starts to attack this myelin sheath here. And when that happens, first this axon becomes dysfunctional, then this axon can be lost, and eventually that neuron is lost. And you start to accumulate neuronal death, and that results in neurologic disability. One very important thing is that we actually have an animal model of multiple sclerosis, which is called experimental autoimmune encephalomyelitis, which is basically induced by immunizing mice with proteins coming out from here, from myelin. That is a model that recapitulates many aspects of multiple sclerosis. Obviously, no animal model recapitulates entirely a disease, but this model has been good enough for us to develop efficacious therapies that actually are now improving the lives of patients. When we go into the clinical course of the disease, right, we can identify at least two important phases. First of all, we have what we call the relapsing remitting phase, and these are what we call attacks, right? These are patients that will come up with blindness in one eye or suddenly some motor dysfunction and so on and so forth. And what characterizes these early phases of the disease is that, the patients will have an attack and they will recover from it, sometimes completely. And that might happen many, many times, and that actually happens for decades. We can actually, with therapies that we have nowadays, really prevent the formation, we can prevent the occurrence of those attacks. But eventually, these patients start to become what we call secondary progressive. And that's basically manifested by two observations. First of all, the course of the disease changes. You stop to see these attacks, well-defined attacks followed up by a clear remission. You start to see these attacks where the remission, the recovery is not complete. And eventually you start to see this progression, right? This accumulation of neurologic disability that is independent of attacks. That's one of the things that characterizes the progressive phases of the disease. The most important thing, though, is that actually we have very good therapies for patients in this relapsing remitting phase, yet we have no therapy whatsoever for the secondary progressive phase of the disease. And that's important because after diagnosis, 10 years after diagnosis, about 50% of the patients will become secondary progressive. Continuamos. Okay, so what I was saying is that there's basically these two well-defined phases of the disease, right? And they are clearly characterized by, first of all, clinical progression, and also a very practical implication, which is response or not to available therapeutics. So how is it that we think this is going on? Do we think that these things are happening? What we think is going on is that there are actually different mechanisms that drive disease pathogenesis at different stages of the disease. In the early stages, right, those that show these well-defined attacks follow up by total or quasi-total remission, what we believe is going on is this is a clear manifestation of T-cell autoimmunity. Basically what we have is T-cells, the same T-cells that fight off microbes and pathogens and tumors. We have them activated in the periphery, and then they get into the CNS, and once these myelin-specific T-cells get to the CNS, they actually cause myelin damage, and axonal damage. And that's important because we have shown those mechanisms and we have really studied those mechanisms really in detail using animal models. And what's most important, based on those animal models, we've been able to actually come up as a community with multiple therapies that interfere with all these different steps. They interfere with the activation and differentiation of these self-reactive T cells, with the ability to migrate out of the lymph nodes and get into the CNS, and even with the ability to be reactivated within the CNS. So all of those stages we can control well. They are good therapies for that, and obviously some of those therapies are also relevant for other autoimmune diseases. The challenge, though, is as I mentioned, patients eventually become unresponsive to those therapies. So what does change in the pathology of the disease? What we believe is that in the progressive phases of the disease, what we have is the activation of cells that sit in the CNS and can drive pro-inflammatory responses themselves. And the two main cell types we're talking about and we're thinking about is, first of all, microglia, which are basically CNS resident macrophages. These are macrophages that migrate into the CNS very early during development, and they sit there and drive inflammation. And on top of that, we believe that there's a central role for a CNS resident cell, which is non-hematopoietic, is a non-immune cell, yet is a cell of the immune system that can play important roles on inflammation. And that cell type is the astrocyte. And in a second, I'm going to talk more about it. So why is that important? Because when these two cells get activated, first of all, they potentiate each other, right? So you have this immune cell, the microglia, talking to these cells from the central nervous system, which is the astrocyte. They talk to each other, and then they can actually promote additional mechanisms of pathogenesis. And those are actually mechanisms that drive directly neurodegeneration. And most importantly, those are mechanisms which we do not really understand how is it that they are regulated. And on top of that, we do not really know how to intervene or block them therapeutically. So just to summarize and move into this talk, basically what we believe is, I told you about this disease we're working on, which is multiple sclerosis. There are two well-defined phases of it. And we believe that in the earlier stages, we have basically an autoimmune response, and an immune response that is being deregulated. And we'll talk more about it tomorrow. But then in the second phase, we have this CNS intrinsic, right, crosstalk between immune and cells of the central nervous system that somehow seem to be potentiating each other and driving inflammation. So, this is like a TV show with commercials, so let's enjoy it. Good. So, with that in mind, right, so what we are very interested in is trying to understand, for example, what are those mechanisms that control inflammation, and in particular those driven by CNS resident cells. And our focus has been on astrocytes. Astrocytes, they got their name because of their shape. They look like stars, right? And these are cells that they were mostly thought in the context of physiology. They were known to play important functions during development. There are specifically important functions associated with guiding neurons and establishing the blood-brain barrier and somehow participating in the maturation of synapses. Top of that, astrocytes also are important sources of lactate and neurotrophic factors that are central for the proper functioning of neurons. Now, many of those homeostatic functions, right, are actually impaired in the context of pathology. And on top of that, we and others have shown that astrocytes can gain disease-promoting activities. They can actually start producing cytokines. Even when there are not immune cells, they can produce cytokines, right? And by doing so, they can activate microglia and other cells in the CNS. They can also actually produce chemokines, which are a family of factors that actually will recruit pro-inflammatory cells from the periphery. For example, you can think about monocytes. And once these monocytes get into the CNS, they can themselves have neurotoxic activity. And on top of that, astrocytes themselves, they can have neurotoxic activities. So there's this balance, right, between homeostatic activities that astrocytes lost in the context of pathology And on top of that, there are these new activities that they acquire in the context of pathology. And obviously, I'm going to discuss it today in the context of multiple sclerosis, yet many of these dysfunctions, right? Decreased homeostatic function, increased pro-inflammatory functions, have been associated to other neurologic diseases, such as not only MS, but also Alzheimer's disease, Parkinson's disease, ALS, you name it. Now, when we go back, right, to these functions that are lost and gained by astrocytes, it's very important to go back in history. And in particular, I would say we can go back more than 100 years ago to the work of the Spanish scientist Santiago Ramón y Cajal, who got the Nobel Prize a little bit over 100 years ago. And when he described astrocytes, he made two important observations. First of all, not all astrocytes are equal. Just using the staining methods he had more than 100 years ago, he was able to tell, look, they look different, right? Like you can look at these well-defined stars, and these, they look more like medusas or something like that. And the second observation he made was actually the fact that astrocytes are highly interconnected. Astrocytes are actually going to interact with these immune cells like microglia. They're also going to interact with neurons. They're going to interact with oligos. And even more, astrocytes will have these kind of like extensions here that are going to reach out to vessels. Hence, through these processes, astrocytes can also sense molecules produced by circulating cells. For example, cytokines produced by T cells circulating through the meninges. So these are very odd observations. And what I'm going to do with you today is basically to go over those observations, to highlight some new developments, to bring some molecular biology to assign function to these observations. And while doing so, also to describe some new tools we've developed that might help us address these and other questions. So this is good. We're going to have breaks. Sorry. Sí, va a pasar. No, es de caminar. Okay. Listo. Okay, so the first thing I want to touch upon is actually this idea that astrocytes are not all equal, right? And how can we actually reanalyze those observations in order to actually identify molecularly defined astrocyte subsets and the pathways that regulate them? And just to put it clear, and this is almost a highlight of the limitations, right? Many of the things we know about astrocyte subsets are based on the characterization of them using single-cell RNA-seq, single-cell ATAC-seq, or even single-cell proteomics. What we don't know, and this is basically a snapshot, right? This is when we go into a brain sample from a patient, from an animal model. These are the different classes we can identify. We still don't know whether these are actually populations, basically cells that were induced during development and somehow get expanded or contracted during disease, or they're actually activation states, basically phenotypes that cells will take in response to stimulation. And that's the reason why I'm going to use the word subset, which is practically meaningless. It basically means and reflects the limitations of the field. We still don't know whether these are induced during development or they have been induced in response, for example, to disease-associated stimuli. Think, for example, in the brain of an AD patient, right? A patient with Alzheimer's disease. Astrocytes are going to respond to beta amyloid. A patient or a brain of a patient with an infection, these astrocytes are going to respond to some microbe-associated molecules, either directly or indirectly. Now, with that in mind, the question is, or the first question I want to share with you, because at the end of the day, it could be that really you don't care about these questions I'm interested in, but you care about the tools. So the first point I want to make is, how do we study the regulation of different subsets, and how can we even identify those subsets? And for years, one of the tools my lab has been very interested is actually the zebrafish. And the reason we've gone through the zebrafish many years ago, this is actually something that was started when we opened my lab, is the idea that zebrafish, right? They are vertebrate. They have a bony skeleton, and that's important. When you compare it to sharks, which have a cartilage skeleton, these are more developed. Yet, they're smaller, and they have a central nervous system and even an immune system that resembles many of the characteristics of other vertebrates, such as mammals, right? So on top of that, what is interesting and what actually drove my attention towards zebrafish many years back is the fact that you can accommodate many embryos, which are completely developed in terms of the nervous system and the immune system, in 96 well plates. Actually, you can have up to 5 in 96 well plates, in each well of a 96 well plate. And then you can use them in order to conduct small molecule or genetic screens. Okay, so for years we've been interested in zebrafish, right? And as I said, what we were interested, or the reason we were interested about them is because we can conduct small molecular genetic screens in order to understand or focus on regulatory pathways associated with any biological process of interest. As long then, obviously, we might have to validate in humans and mice to see that whatever we find in zebrafish is relevant. So a few years back, what we did is we actually developed a novel model of neurodegeneration in fish, in zebrafish, neuroinflammation. And those studies led us to identify a novel signaling pathway by which a ligand-activated chaperone that sits in the ER, which is called sigma-1, we found that actually was able to control the unfolded protein response. And specifically, we found that this specific signaling pathway was able to trigger the activation of this transcription factor, of a transcription factor called XBP1. We thought that was interesting, and actually what we found most interesting is that actually this transcription factor, right, if we now go and inactivate it specifically in astrocytes, we can really ameliorate this experimental model of multiple sclerosis. And this is just, since this is the first time I'm going to show you data in this model, basically in day 0, we actually induce disease by immunizing these mice. Control mice show a significant worsening of EIE, right? Like we can see how we have an accumulation of clinical score. But if we now specifically inactivate this transcription factor XBP1 in astrocytes, or also if we inactivate the enzyme that activates it, we get a significant amelioration of the disease. And interestingly, if we now sort these astrocytes and analyze their transcriptional responses, these, when we inactivate these transcription factors, there's a significant downregulation of genes associated with the pro-inflammatory, disease-promoting activities of astrocytes. We thought that was interesting. The other thing that we found interesting is that even if we only inactivated this transcription factor in astrocytes, we could still downregulate the pro-inflammatory responses of microglia and monocytes, suggesting that this transcription factor not only controls cell-intrinsic mechanisms of disease pathogenesis, it also controls the ability of astrocytes to induce pro-inflammatory responses in astrocytes and microglia. And this cell-cell communication is going to become important later on. So with that in mind, okay, this is very interesting. We found it in zebrafish. It works in mice. What about humans? So we actually went to and analyzed CNS samples, brain samples from multiple sclerosis patients. And to cut it short, we found a significant upregulation of the pathway, right? This XBP1-driven signaling pathway in lesions in multiple sclerosis patients. So this even suggested that this is relevant not only for mice and animal models of multiple sclerosis, but actually for the disease itself. Well, okay, perfect. Continuamos. So we identified these astrocyte subsets, right? That is expanded in MS tissue. So we thought, okay, this is great. Let's go and really go in depth into this astrocyte subset in mice. And let's try to really understand what are the molecular mechanisms that control it. So the first thing we did is actually we use the very same antibodies we have used to characterize it in humans. We go to the animal model, right? We go to the EIE. And then we see a nice upregulation of these astrocytes characterized by XBP1 increased signaling in the spinal cord and the brain of mice, right? And we also see it of sick mice. And we see it both in the gray and the white matter. So we thought this is great. Let's just go and do single-cell RNA-seq and get all smart and learn about what regulates them. So if you remember, just to remind you, What we need to be able to detect is a very tiny thing. It's basically the activation of this transcription factor called XBP1. But this is a tricky activation, because the way it works is that this transcription factor is actually going to be transcribed most time harboring an intron that has an early stop. So basically what that means is that unless this pathway is activated, you're going to have a transcript that encodes a non-functional short protein. Now, if the pathway gets activated, there's a specific enzyme that gets activated that catalyzes the alternative splicing. This early stop in the intron, and this intron with the early stop is removed, and then suddenly you encode a functional transcription factor. So we thought that would be easy. We just sacked some mice and did some RNA-seq, and that's literally when travel began. Of course, we sequenced, for example, about 20,000 cells from the CNS, and out of those, about 1,000 were astrocytes. Yet, when we tried to detect the expression of these molecules here, we only detected it in 14 cells. Now, when we go with immunohistopath, right, we can detect them by immunofluorescence. So this is basically telling us there's some limitation in the RNA-seq, in the single-cell RNA-seq we're running, that doesn't allow us to detect these specific cells. So that actually triggered quite some efforts in the lab, which is because this is a problem that us as scientists have many times. We can be interested, or we might be focused on a specific population, which is located in a specific area, which we know is physiologically relevant. I showed you if we knock them out, there's a significant effect in disease. Yet, we cannot enrich for those cells. So there are two options, right? We can either sequence a lot of cells and then hope that if we have the money to enrich, to sequence lots of cells, we're going to detect enough data. Or we might be able to detect, or we might have to develop methods to detect and sort those few cells which are really important to us. And this is what we wanted to do. And in order to do so, we developed a technique which we call find-seq. And this is basically a technique which is based on co-encapsulating your cells of interest with agarose beads. And those agarose beads, what they are going to do first is we're going to use them to capture all the RNA and all the DNA of your cells co-encapsulated there. We're then going to produce or synthesize the cDNA there, and then we're going to run a reaction which is called, which is basically a digital PCR. It's a PCR where the amplification of your gene of interest or your genes of interest, because we can do it in multiplex, is going to result in that specific droplet expressing in one specific color. For example, that specific droplet becoming green. Why is that important? Because then we can sort for droplets that have expressed the genes of interest. We can only sequence those droplets, and then we can really enrich or focus our efforts on sequencing those cells we're really interested on. On top of that, since we're actually capturing from the very beginning the RNA and the DNA on those agarose bits, that's actually going to protect that material from degradation, and that's actually going to result in better reads, in deeper sequencing results. So let's see how it works. So we went back to our original problem, right? How is it that we can analyze and focus on those cells of interest? So in order to analyze that, we induced EIE in mice, we sacked them at the peak, and then we focused on cells expressing aquaporin, which is a classic marker of astrocytes. And on top of that, cells that also expressed this gene called EDEM1, which is a classic marker of XBP1 activation. This is the data you get here. This is very similar to what you've seen thousands of times, like flow cytometry. There's only one difference. The signal you get here, it's not signal produced by antibody binding. The signal you see here is actually the signal produced by this digital PCR, where you're actually amplifying your gene of interest. This is actually a qPCR for aquaporin 4. This is a qPCR for EDEM1, which is a marker of XBP1 activation. So now we can see the different populations. This increase in the double positives, basically this increase in astrocytes that show an activation of this pathway called XBP1, recapitulates our previous observation in immunofluorescence. And now we can actually, what we can do is we can sort each one of these cell types or cell subsets, same way you do FACS sorting, and we can actually go deep and characterize the transcriptional profiles. So we did that, and the first thing we did was actually some kind of sanity check. First of all, we did detect a significant pro-inflammatory response in these astrocytes, and that's not surprising, because that's why we were interested in this population, right? Moreover, when we look at the predicted transcriptional regulators of this population, one of those we picked up is actually this gene called XBP1. So to cut it short, basically we are isolating and analyzing what we wanted to. So now the question is, what else? What new biology can we learn by using this tool? So we actually looked at the new data set we generated and identified a whole set of candidate positive and negative regulators of this pro-inflammatory astrocyte subset. You can see them here. Obviously, XBP1 is still there. And one of the things we wanted to do was then, how can we test which one of these is functionally relevant? So in order to address that problem, what we decided to do was an in vivo CRISPR screen. So basically, we generated a virus where we can actually inject with the virus a guide RNA and the CRISPR machinery and make it express specifically in astrocytes. And we inactivated each one of these gene candidates in astrocytes. We test the functionality of each one of these candidates. So as I said, we inactivated each one of them in vivo, and then we induced EIE. And as you can see here, most of them showed no phenotype whatsoever. When we inactivated most of them, there wasn't really big differences, right, in terms of EIE development. However, one of them, actually the gene encoding the mineralocortical receptor, when we inactivated it, that resulted in a significant upregulation of the disease, a significant worsening of the disease, suggesting that mineralocortical receptor signaling within this specific astrocyte subset somehow promotes, somehow blocks, right? It's a break, limits pro-inflammatory activities in this astrocyte subset. And just as a note, because I'm going to skip mechanisms in order to go a little bit faster, one thing to keep in mind is that the mineralocortical receptor can act as a transcription factor, but it can also control the epigenetic status of target genes by interacting with other cofactors. So in order to understand a bit how this transcription factor, right, how this mineralocortical receptor was involved in suppressing the pro-inflammatory activities of astrocytes, in particular those astrocytes driven by XBP1, we sorted those astrocytes and performed sequencing. And to cut it short, what we were able to show is that actually the mineralocortical receptor controls the epigenetic accessibility of many of these pro-inflammatory genes. Basically, it inhibits the ability of transcription factors to be recruited to the regulatory regions of these genes and transcribe them. And among those genes targeted is actually this gene, which is XBP1, which is the one that seems to be driving the differentiation of those pro-inflammatory astrocytes. The last thing we wanted to do was actually to see how relevant this could be for humans. And in order to do so, we actually analyzed a data set of about 50 single cell and single nuclei data sets from individual patients, MS patients, and controls. And to cut it short, what we notice is that in MS patients, there's a significant downregulation of the mineralocortical receptor and its signaling machinery in astrocytes in MS patients, and that seems to be negatively correlated with an increase in pro-inflammatory responses. So to finalize this first vignette of what I wanted to share with you today, first of all, I told you about how we're interested in trying to understand few cells, a small subset of cells that seems to have an important role in CNS pathology. And in order to do that, we developed this new technique, which we called FIND-seq, which seems to be important for the development with this. Okay, so just to summarize this first struggling section of the presentation. So, first thing is, so I show you about the specific XBP1-driven population of astrocytes that seems to be contributing to disease pathology. I didn't have time to show you, but basically what we show is that the mineralocorticoid receptor, which now we identified as a negative regulator of this astrocyte subset, interacts with NCOR2 in order to really control the accessibility of these astrocyte subset. I'm sorry, just getting emails that are not mine. This is not my computer, so I'm not responsible for this. Socios y accionistas. Esto es interesante, pero no tiene nada que ver con lo mío. Okay, so, and then the second thing I wanted to bring up is the idea of this new tool we developed that basically kind of solves, or we think allows you to solve a big problem, which is all of us, right, where we come up with our single cell analysis of whatever cell type in whatever tissue, but most times either those cells are too rare or we don't have surface markers in order to isolate and reach for them and go really deep in their characterization. So what I'm showing you today is basically a tool which we call FindSeq that allows us to characterize cells of interest just focused on the expression of nucleic acid markers of interest. And why do I use the word nucleic acid? Because in the work I show you here, we focus mostly on astrocytes and using RNAs, messenger RNAs as markers. In work we have done with folks at NIH, Danny Dueck, Eli Boritz, and others, we actually use this very same approach to analyze frozen samples from patients which have a chronic HIV infection and used actually HIV sequences integrated into the DNA as a way of sorting out cells of interest and then trying to understand why is it that the virus can establish latency there. So this is the first thing I wanted to share with you. And now I want to go into a larger question, which is obviously this is only one of the subsets we and others have characterized. And the last piece of information I want you to leave is that the same way that we characterize this subset, there are multiple subsets, multiple activation states or populations of astrocytes that seem to be contributing to disease pathology. We have characterized many of them by combining proteomics, single-cell proteomics, single-cell genomics, and in vivo perturbation states. And just to give you a flavor of the different colors or shapes or functions that astrocytes can play in inflammation, first of all, we characterized not so long ago a subset of astrocytes which is pathogenic and is driven, for example, by a transcription factor called MAFG. And the point I want to highlight, because it's going to be interesting, is that when we look at who induces it, where are these astrocyte subsets coming from, we noticed that it's actually coming from cytokines produced by T cells that infiltrate the CNS. So again, going back to that old idea from Ramon y Cajal, which is the idea that astrocytes are highly interconnected with other cells, and suggesting that those connections can really control astrocyte fate and function. And the same way, we have pro-inflammatory astrocyte subsets. More recently, we characterize anti-inflammatory astrocyte subsets, which seem to line the borders of the brain and whose function seems to induce T cell apoptosis, basically to prevent T cells from getting into the CNS. And again, and just to finalize, what I wanted to highlight, this is an anti-inflammatory astrocyte subset, and the main reason I'm showing these two extremes is the idea that there are multiple functions or subsets or functional states that astrocytes can take in the context of inflammation, neurodegeneration, to really promote or limit inflammation. The last point I want to make about this specific anti-inflammatory astrocyte subset is that it's also induced by this communication between astrocyte and another cell type. In this specific case, it's mediated or driven by astrocyte interactions with natural killer cells that circulate through the meninges, and as they circulate, they produce cytokines. So this actually is bringing us back to this very old concept, right? This very old drawing from Ramon y Cajal, the idea that astrocytes are really highly interconnected, but now we're starting to see how those interconnections control fate control function. And if we just have a very brief summary about connections established with astrocytes between other cell types, right? We do know, and I just mentioned briefly, interactions between astrocytes, T cells, and NK cells. We also know that astrocytes, as I mentioned earlier, they can produce trophic factors, they can produce metabolites that really control neuron function, and they can also control oligo function. And one of the best characterized interactions are interactions mediated with between astrocytes and microglia, which are the CNS resident macrophages. And these are some examples from our work where we characterize some of those interactions. The problem, and I'm starting from our own work, is that the limitation of many of those works, right, is that you're working on one specific problem, and then you try to identify what's the specific mechanism, right, that mediate that interaction between astrocytes, T cells, microglia, neurons, whatever. We're still lacking a method that would allow us to study cell-cell interactions between astrocytes and other cells in the central nervous system, or you might even think about your favorite cells and other cells within the tissue in an unbiased manner. And that's not a trivial problem because, as Ramon y Cajal showed many years back, those cell-cell interactions are central for basically tissue homeostasis and pathology. So the question we wanted to address is, how is it that we can study cell-cell interactions in an unbiased manner? And in order to do that, we developed a technique which we call rabbit-seq. And the reason we name it like that is because it's based on rabies, the rabies virus, which historically has been used to map cell-cell interactions. So the way rabbit-seq works is basically we actually took the rabies virus, we modified it. The first thing we did is we pseudotyped it, so now we can infect any cell of interest. And on top of that, we actually incorporated mCherry. So that's basically a fluorescent protein, so we can actually know the cells which is infecting. But we actually tagged that mCherry with a barcode, with basically a gene barcode, a genetic barcode. And why is that important? Because for the studies I'm going to show you now, we didn't use one single modified rabies virus. We actually used a library of rabies viruses, each one of them having a different barcode. And how does that work? Basically, the virus is going to first infect astrocytes, right? And then it's going to jump only once because of another set of changes we introduced to cells that are interacting with those astrocytes. And since we have not only one single virus, but actually a family, a whole library of millions of barcodes, we can actually study multiple astrocyte microglia, you name it, interactions in parallel to try to identify common elements, to try to identify specific interactions that are affected in the context of pathology. So let's see how that works in real life. So the first model where we apply that is the EIE model I showed you a couple of times now. So we induce disease, we introduce our virus library, we isolated the cells infected because they express mCherry, and then we did single cell sequencing of those cells. And the first piece of data you get is something you've seen a million times. It's basically a t-SNE plot, a UMAP, where each one of those dots represents a cell for which you have single cell transcriptional data. The only difference, what makes rabbit-seq different, is that each one of those dots, basically each one of those cells, is annotated with a unique genetic barcode which has been introduced by the virus. So now we can use that barcode and actually the sharing of the barcode among different cells to organize a UMAP like this into an interaction plot like that. So now we can start to reconstruct the cell-cell interactions that each one of those cells established in vivo. And since at the end of the day, all of this is based on single-cell transcriptional data, we can actually focus on specific astrocyte subsets or specific microglial subsets involved in those interactions and try to ask some very interesting questions. First of all, how do cell-cell interactions change over time, right? When you induce a disease. And this is a clear example. If you look, for example, in a naive mouse, astrocytes are mostly interacting with microglia. Yet, if you induce inflammation in those mice because you induce EIE, suddenly you start to detect interactions with other immune cells, such as T cells, dendritic cells, so on and so forth. First thing we can do is like to see and to detect what interactions are gained or lost in the context of pathology. But then, since we have at the end of the day single cell data between interacting cells, we can really try to identify the mechanisms that mediate those interactions and what is the effect of those interaction mechanisms on cellular responses. And indeed, that's actually what I'm going to show you. We actually apply that very same approach in order to try to study a very basic question. What are the mechanisms that mediate interactions between astrocytes and microglia? Can we find something new? And in particular, since we have single cell data, we decided to be very smart and say, let's focus on those astrocytes that because of their transcriptional signature, they show the most pro-inflammatory response, right? We can find transcription factors such as NF-kappa B and others that are really pro-inflammatory. So we first identify those astrocytes, we identify them in our rabbit-seq data set, and then we look backwards and we looked at the microglia interacting with them. Can we identify novel mechanisms of interaction that really promote those pro-inflammatory responses in astrocytes? And we did that, and to our surprise, what we found were not actually mechanisms of cell-cell interaction mediated by cytokines. That was actually what I was waiting to find. What we found was the reactivation of a set of pathways which are called action guidance pathways. And without getting much into the details, basically these are mechanisms of cell-cell interaction that operate during CNS development. Usually not between astrocytes and microglia, but for example, between neurons and astrocytes, neurons and other cells. So our data suggested that these mechanisms were somehow co-opted or misactivated between astrocytes and microglia in the context of pathology. And among those pathways, the one that was leading our scores, right, the strongest candidate, was a pathway which involves a protein, a receptor called the FB3 receptor, and its ligand ephrin B3. And there's something interesting about this pair. First of all, the FB3 receptor is actually a tyrosine kinase. So that means that if this interaction turns out to be important, then we can try to design kinase inhibitors in order to block it. On top of that, what we found is that this interaction was mediated by the ligand called ephrin B3, which first of all is membrane bound, and it's not only membrane bound, it also has an intracellular domain that can trigger signal transduction. So the practical implication of that observation is that interaction mediated between these two cells, between microglia and astrocytes, mediated via FB3, ephrin B3 signaling, can actually have bidirectional effects. They can basically modify the cellular responses of astrocytes and microglia. And we thought that would be extremely important because at the time, those interactions have not been described. So obviously what we wanted to do, this is kind of like the first outcome of kind of a screening campaign. How can we validate those? So the first thing we did was actually to go back to MS tissue, MS lesions from MS patients and controls. And to cut it short, what we detected was a significant upregulation of the expression of the receptor FB3 in astrocytes located in MS lesions, and a concomitant upregulation of its ligand in microglia located in those very same lesions. Indeed, if we actually analyze single-cell RNA-seq data from these cell types, we detected significant signaling associated with the activation of these pathways. Based on that, we decided to interrogate those findings in functional genetic perturbation studies. So the first thing we did was actually to inactivate either the FB3 receptor specifically in astrocytes or its ligand in microglia. We then induced disease, and as you can see here, there's a significant amelioration of EIE, there's a significant decrease in monocyte recruitment to the CNS, and a significant decrease in astrocyte pro-inflammatory responses. So to cut it short, these interactions between astrocytes and microglia mediated via FB3 expressed in astrocytes and ephrin B3 expressed in microglia, were somehow triggering a pro-inflammatory signaling in astrocyte, a pro-inflammatory signal in astrocyte that was triggering this pro-inflammatory responses. So now to understand in detail the mechanisms by which this signaling pathway triggers pro-inflammatory responses in astrocytes, we actually went back and performed a series of transcriptional analysis. I don't have the time now to go over them, but to cut it short, what we found was that signaling mediated by the tyrosine kinase in this FB3 receptor controlled mTOR signaling, and by doing so, controlled mitochondrial function, and in particular, controlled the production of mitochondrial reactive oxygen species and other mitochondrial products that can trigger pro-inflammatory responses in astrocytes. And I can tell you more about that during the Q&A. Then, as you remember, this is a signaling pathway that is interesting because it's bidirectional, right? So also the microglia could see their responses modified as a result of these interactions. And to cut it short, what we found was that these interactions trigger actually and boost NF-kappa B signaling in microglia. And to be honest, we don't know specifically what are the specific mechanisms by which signaling initiated in this intracellular domain results in NF-kappa B signaling. That's something, or in exacerbated NF-kappa B signaling. That's something that we're trying to figure out. So we found this pathway, we identified this pathway. It seems to be pro-inflammatory. We were able to validate it both using not only mouse systems in vitro and in vivo, but also using human astrocytes and microglia in vitro. So the last question we wanted to address was whether we could somehow target it therapeutically. And in order to do so, we basically exploited the fact that the intracellular domain of this FB3 receptor is actually tyrosine kinase. And our studies have shown that that tyrosine kinase is essential for the ability of these interactions to promote CNS pathology. So we perform a small molecule screen. We perform a small molecule screen of using several libraries. In particular, we had a library of CNS penetrant molecules, right? Because obviously what we want is to come up with small molecules that can cross the BBB and interfere with that specific interaction within the CNS. So when we performed that, we identified one molecule that we called A38, and we found that this molecule was able to, first of all, suppress A38 and basically FB3 signaling in vitro and in vivo. And most importantly, when activated following disease induction, it had the ability to ameliorate EIE, and it had the ability to suppress many of astrocyte pro-inflammatory responses. And we showed that not only in this, but also in additional models of multiple sclerosis. So then to summarize what I wanted to share with you in this section of my talk, so we developed this technique, which we call rabbit-seq, which enables the investigation of basically cell-cell interactions in vivo. And that's important because initially we were limited by the need of specific transgenes being expressed in the mice when we were performing these studies. Why is that important? Because we're using this model, among other things, right? This tool, among other things, in order to identify mechanisms of disease pathogenesis, for example, relevant for MS. So we identify a mechanism in mice, and then we go and try to validate it with humans. And one of the challenges we face is it could be great if we can actually change the paradigm. What if we can change this technique so we can actually apply it to investigate clinical samples, and then whatever hit we identified, we actually then go on and study it in depth in animal models. So to cut it short, we were able to modify this technique so it works in clinical samples. And in particular, we use it to understand how brain tumors talk with other cells, among them astrocytes in the CNS. I won't get into the details. The reason we started with glioblastoma was because actually, as you know, one of the first interventions to which patients are subjected is actually a removal of the tumor. So that gave us a good supply of clinical tissue for us literally to optimize our approach. So we were able to identify, and I won't go over this now, multiple targets, basically mechanisms by which the tumor talk to astrocytes and other cells in order to evade the immune response. And then we were able to actually validate them in animal models. And we believe that has led us to identify, some of them did not turn out to be very good, but some of them are actually what we believe good candidate targets in order to actually increase responsiveness, actually improve the efficacy of immunotherapies in glioblastoma. Why is that important? This is actually one of the most lethal tumors where the mean survival after diagnosis is 14 months. That's why there's such an important need to really understand how is it that the tumor can evade the immune response. But on top of that, now we developed this technique, we can actually go back and use it to analyze other clinical samples we obtained from multiple sclerosis patients. Actually, we were able to use this technique in order to study monkeys and aging monkeys as a way to understand how is it that neuroimmune communication is perturbed as we age. And we were also able to use this very same approach as a way of studying how is it that neuroimmune interactions are affected in the gut, for example, in the context of inflammatory bowel disease. Super. So now to summarize this part, so basically what we did is I told you about this new technique, and then the very, which allow us now to study clinical samples and so on and so forth. But then there are very factual, very important follow-ups or outcomes, among them the identification of a new pathway that mediates cell-cell interactions between astrocytes and microglia and promotes neuroinflammation. That's important because, as I mentioned, this is a pathway that we haven't thought of in terms of its role in neuroinflammation. And most importantly, we identified a small molecule that can interfere with that pro-inflammatory signaling. And we have a very active a medicinal chemistry program where we're really optimizing this technique, this molecule, with the goal of putting it into the clinic. And then to get into the last part of my talk, and basically is the idea of, every method we come up with has its limitations, right? You could argue, we apply this rabbit-seq technique, and you could say, well, probably the interaction I'm looking for is not deregulated in that specific model. It's probably not that deregulated at that specific time point. Wouldn't it be great to come up with a method to basically have a catalog of every single mechanism mediating the interactions between your two favorite cells? And then once you have a catalog, right, once you have your candidates, you can go all smart, right? You can go to clinical samples and you can try to validate it, or you can perform perturbation studies in vivo. So in order to address that need, in order to address that problem, we develop a technique which we call SPEAK-seq, which is basically a platform in order to perform what we call forward genetic screens to really identify every single molecule mediating interactions between your two favorite cells. We first applied this technique to study again the interaction between microglia and astrocytes. And the way it works is relatively simple. We basically developed a method to culture cells in droplets for up to 3 days, but actually we do not culture one single cell. In each droplet, we have 2 cells. One cell, actually in this case microglia, harbors a genome-wide CRISPR library. So in each droplet, we inactivated one specific gene. And the other cell, in this case, is an astrocyte with a reporter. So basically what will happen is every time we perturb an interaction, right? We perturb a gene here in the microglia that is important for the ability of microglia to talk to astrocytes, then that astrocyte's receptor is going to light up. So that's basically what we developed. Obviously, there are a lot of technicalities that have to be solved up. Obviously, it has to do with growing cells in droplets and also developing microfluidic devices where to co-encapsulate those cells and where to sense, first of all, the presence of each one of those cells, because we only want to focus on cells that have on droplets that have 2 cells, right? That's important. But on top of that, to be able to sense the activation of this specific reporter. So we did that. And then when we apply that approach, the first thing we come up with is something like that. This is the first time we have a catalog of every single gene that could potentially mediate the interaction between astrocytes and microglia. That's exciting, right? Because now we can start we can go over databases and we can try to understand what is the function of those genes. In particular, and what I'm going to show you today, we focus on 4 candidates which we thought were exciting. These are ligands produced by microglia, and these are its receptors expressed in astrocytes. So we had these 4 candidates. The question is, how can we know which one of them is relevant, again in this case, to our model of CNS inflammation, which is EIE? And in order to address that question, what we did was we used an approach which is called Perturb-seq, where we basically introduced a library of guide RNAs into the CNS of mice. Each one of those RNAs had a barcode, and basically we looked, and each one of those RNAs obviously was targeting one member of these signaling pathways. And the question we address is very simple. Which one of these pathways, when activated, results in the highest deregulation of astrocyte responses? Those studies led us to identify as a candidate this pathway, which involved the secretion of a protein called amphiregulin, signaling via a receptor called the EGFR. So just a bit of background. What is amphiregulin? Amphiregulin is actually a protein that has been described to be very important for the maintenance of epithelial barrier and tissue in the gut. For example, in the context of inflammation, it has been shown to be very important for promoting that gut and epithelial barrier homeostasis. There was one report suggesting that actually amphiregulin could involve or could mediate the regulation of astrocytes. That was in the context of stroke. And interestingly, that report suggested that regulatory T cells, and tomorrow I'll talk more about regulatory T cells, that are recruited to the CNS in the context of stroke, seem to deactivate astrocytes via the production of amphiregulin. To cut it short, actually, we generated a conditional knockout of amphiregulin in T cells, and we saw no phenotype whatsoever on EIE. So we believe that this observation probably does not apply for models of autoimmune inflammation, such as multiple sclerosis and EIE. And the last piece of information I want you to hold on to is the fact that IL-33, a cytokine produced by cells under stress, that's why it's called alarming, has been shown to be an inducer of amphiregulin, for example, in the gut. And we'll get back to that. So this is what we knew about it. And the first question is, how can we validate whether this pathway is relevant or not? So the first thing we did was actually to CRISPR out amphiregulin from microglia. And as you can see here, when we do that, there's a significant worsening of EIE. So that is literally telling us that amphiregulin produced by microglia seems to act as a brake to suppress astrocyte pro-inflammatory responses. And indeed, if we actually sort those astrocytes and analyze the transcriptional profile, when microglia cannot produce amphiregulin anymore, that results in a significant exacerbation of astrocyte pro-inflammatory responses. So that suggested that amphiregulin produced by microglia acts on astrocytes in order to regulate or to suppress the pro-inflammatory responses. Then the last question we wanted to address is, how is it that amphiregulin is regulated? How is it that microglia knows that it has to produce amphiregulin? And for that, we went back to our rabbit-seq dataset, the dataset we generated a few slides ago, and we basically focused on microglia producing amphiregulin. What were their characteristics? What were their upregulators? Their pathways regulating them. And what caught our attention was the fact that we detected a significant signature associated with IL-33 signaling. As I mentioned, IL-33 is actually a cytokine that has been described in other cell subsets to induce amphiregulin production. So we postulated at the time that potentially amphiregulin could be induced by IL-33 produced by astrocytes. What these data suggested is that potentially IL-33 produced by astrocytes could be somehow telling microglia of the need to produce amphiregulin to arrest pro-inflammatory responses. So how could we validate that? So first of all, we generated a conditional knockout mouse where we inactivated the where we inactivated the IL-33 receptor in microglia. And as you can see, when we do that, we detect a significant worsening of EIE. Indeed, if we now take microglia in vitro and throw IL-33 onto them, they will produce microglia. And now if we do the opposite, if we just go and generate astrocytes which lack IL-33 production, then these astrocytes cannot produce IL-33, and then that results in a worsening of EIE, and indeed we do not induce microglial amphiregulin. So to summarize what I showed you before, and then just to start closing on what I wanted to share with you today, first of all, we identify what we think is a nice neuroimmune circuit, right? This is a cell circuit which involves astrocytes being stressed in the context of inflammation, secreting IL-33, and that signal being sensed by microglia as a way of inducing the production of amphiregulin to somehow deactivate that pro-inflammatory response. And the second piece of information or take-home message I want to leave with you is the idea of developing this novel platform, right, that basically allows you to perform CRISPR-wide, genome-wide CRISPR screens to really identify all molecules involved on cell-cell interactions of interest. And obviously then it's up to you to validate them in the best preclinical and clinical models. And with that, we go back to where we started, right? And the take-home message I want to leave is this idea that astrocytes are not equal. I show you data in terms of how they can be pro or anti-inflammatory. And most importantly, I showed you how their interactions with other cells, either immune cells resident in the CNS, such as microglia, or other immune cells recruited to the CNS, such as T cells and NK cells, can play important roles in the control of astrocyte responses and also in the control at the end of the day of CNS pathology. So just why is that important? Because as immunologists or neuroscientists, that brings us back to really study these two systems that, we tend to study and understand in isolation, the nervous system and the immune system, which actually shared many common characteristics. And just to bring one example, both of them are the only systems that can establish memory. Obviously, we have memory from a cognitive point of view driven by neurons, but also we have immune memory, which is at the basis of us having efficacious vaccines. These two systems cross-talk a lot in pathology, but also in the context of homeostasis. That's important for memory, for the control of behavior, for the control of CNS development. So why is that important? Because what we believe is that these tools we have developed can help us understand the communication between those two systems. And in particular, what we are trying to do, and what I think it would be a good challenge for the research community, is actually to take forward a concept that is well known and deep into neuroscience. Neuroscientists for years have been talking about the connectome, the idea of really trying to build and to understand any possible interaction, right? All the interactions between all neurons in the CNS and how they are associated, and the perturbations are associated with behavior, how those perturbations are associated also with disease. And we can think, for example, you might have heard about how the autistic brain can be wired differently. What we want to do is actually to use many of these tools we have developed actually to build what we call the neuroimmune connectome, which is going to be different from the classic connectome that we think about neurons, right? Neurons are actually next to each other. The neuroimmune connectome is actually much more fluid. It's mediated by soluble factors. It's mediated by immune cells that could be patrolling the CNS. However, we do know that neuroimmune connectome is central for our ability to understand not only CNS development and homeostasis, but also CNS pathogenesis. And that's what I believe most exciting, because if we really understand how those two systems talk to each other, we really can come up with novel therapeutics. We can use, for example, small molecules, right? I was talking about this A38 molecule that can allow us to really target specific interactions and try to somehow reestablish normal and healthy neuroimmune interactions. But on top of that, we can think about wilder things. What about developing synthetic probiotics, right? Or synthetic microorganisms, either yeast, bacteria, or viruses, to really target those perturbed neuroimmune interactions and have a new set of therapeutics. And this is actually something that probably I'll tell you more about tomorrow if this thing works properly. And with that, I'm very happy that we finished. I'm going to acknowledge the work of all of those in the lab that really ran these studies, in particular, Fibrod Hammer, Jessica Kenninson, Mac Wheeler, Hong Lee, and Ian Clark are the ones that really developed many of the tools and techniques that you've seen here. And I'm now very, 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 very happy to take any questions you may have.
Thank you very much. ¿Alguna pregunta que tengan? ¿Me ayuda alguien con el micrófono, por favor? Ustedes dos. Eduardo, ¿tiene alguna pregunta alguien?
Gracias. Should I do the question in English or?
No, puedo hablar casi normal en español.
Okay, muy bien, gracias. Entonces, han mostrado que realmente como pues primero, gracias por la presentación, está fantástica y también todos los datos son llamativos de una trayectoria de trabajo que fue como muy larga y muy intensa realmente. Entonces, tal vez es un ejemplo para nuestros estudiantes y tal vez unos profesores, ¿no? Tal vez, como yo. Pues primero, dos preguntas. La primera es si su modelo de esclerosis múltiple recapitula los eventos temporales de la esclerosis múltiple donde hay una recuperación, por ejemplo, de los síntomas. Y tal vez podría expandir un poquito en eso para justamente cuando usted hace la determinación de los contactos, por ejemplo, célula-célula entre asocitos y microglia, ha identificado justamente estas parejas moleculares que son también comunes en neuronas. Entonces, ¿cómo usted recapitulan justamente, cómo usted realmente ahorita ponen en el juego, reponen en el juego ahorita las neuronas, ¿no? Porque el sistema era caracterizado realmente como enfocado en astrocitos, microglia, pero la parte neuronal que comparten estos determinantes moleculares, ¿cómo ustedes lo reponen, ¿no? En el sistema? Pero primero tal vez el modelo, cómo sus similitudes, por ejemplo, con la enfermedad.
Perfecto. Hay muchos modelos que recapitulan distintos estadios de la enfermedad, ¿cierto? Entonces, en este caso en particular, nosotros nos enfocamos en mecanismos que son más relevantes para las fases progresivas de la enfermedad y en particular para la desregulación de la interacción entre astrocytes y microglia, right? Entonces, la pregunta es qué rol juega, y en ese sentido el modelo es válido, y por otro lado yo les mostré, utilizamos otros dos modelos para aún further validate these findings. Ahora, existen además, la pregunta es qué rol juegan las neuronas, y en realidad esa es una pregunta mucho más grande, porque esas interacciones, esas neuroimmune interactions son importantes no solo para la biología de la esclerosis múltiple, sino también, por ejemplo, para behavior, para memoria y un montón de otro tipo de cosas. Ese es actually la línea de investigación que Mike Wheeler, que fue un postdoc en el laboratorio, ahora está desarrollando en su propio laboratorio. Así que capaz en unos años lo vas a escuchar de él, pero eso es lo que viene. Por otro lado, en el contexto de esclerosis múltiple, los nuevos datasets que estamos generando a partir de pacientes, en ellos cambiamos la técnica para capturar las interacciones con neuronas. Y al principio, el no incorporarla fue una decisión estratégica. Cuando recién estamos poniendo a punto la técnica, la mielina y el FICOL era extremadamente sucio para después nosotros establecer las libraries. Entonces, we actively left it out. Con el tiempo logramos optimizarlo y entonces los nuevos datasets que generamos de humano y de pacientes con MS tienen las interacciones con neuronas y hay una postdoc que está empezando el 1 de agosto para estudiar eso. Creo que es interesante también desde el punto de vista bidireccional, porque en MS nosotros tenemos a pensar las neuronas como el target del immune response, pero por otro lado, productos de neuronas pueden tener efectos significativos en la inflamación. Y entonces hemos identificado algunos de esos y ahora lo que nos queda es tratar de identificar por qué, cuál es el mecanismo involved.
Tal vez no sé si puedo robar otra, otra pregunta, porque sí es tan fascinante como el trabajo, pues la enfermedad, entonces la esclerosis múltiple, pues la ha mostrado de manera experimental, pero recientemente no se ha identificado que tal vez el virus Epstein-Barr puede justamente como tener justamente este hallazgo, ¿no? Puede realmente como llevar a eso. Entonces, ¿cómo ustedes realmente cómo hacen esta dualidad? O no sé si hay como similitudes entre las dos enfermedades o no sé,
Bueno, entonces,
los modelos.
Lo que se sabe, para dar background a todos, lo que se sabe es que hay una mayor incidencia de Epstein-Barr infection en pacientes con MS. A la edad de los adultos generalmente es menor, porque casi todos nosotros tenemos el Epstein-Barr virus, pero si vos te fijas en MS pediátrica, hay una diferencia significativa del 30% versus 80%. Y hay un estudio muy interesante que fue publicado por Alberto Asquerio en Science el año pasado, que mostró, usando un cohort de, creo que era US Army Soldiers, que mostró que la infección por Epstein-Barr en algunos aparentemente precede el desarrollo de MS. ¿Cómo interpretamos esos datos, cierto? La forma que yo y creo que la comunidad lo interpreta es el punto de vista que en gente que tiene el genetic background apropiado, factores ambientales pueden hacer el trigger para generar respuestas inflamatorias disreguladas. ¿Está bien? Y la pregunta, y que de hecho eso ni lo toqué, pero mostré los datos, es cómo estudiamos eso desde el punto de vista experimental. No tenemos métodos para estudiar en un unbiased manner el efecto de factores ambientales en pathways of interest. Todos podemos hacer epidemiologic studies, but for that you need a candidate, right? Entonces, el motivo por el que desarrollamos ese modelo de zebrafish fue una colaboración con la EPA, the Environmental Protection Agency, y lo que hicimos fue, hicimos un screening de about 10,000 different environmental factors to see which one of them could worsen CNS inflammation. De esos factores, uno que es linurón, que es un herbicida, lo que vimos es que activaba este specific signaling pathway driven by XBP1. Entonces, ese creo que es la forma a seguir. Necesitamos, creo que, experimental models que nos ayuden a entender cómo es que el environment y environmental factors can trigger, not only trigger, but actually boost inflammation.
Gracias.
Muchas gracias por esta espléndida charla, abrumadoras, cualquier cantidad de datos que no se tocaron, que están allí. Y muchas gracias por la paciencia. O sea, estas interrupciones, pensé que en algún momento iba a tirar todo y a dejarlo ahí. Pero bueno, el viaje de Boston lo amerita. Gracias, verdaderamente muchas gracias. Mi pregunta va dirigida, primero creo que de alguna manera la acabas de responder, que sería, ¿cuál sería el disparo del estrés proteico que enciende justamente el factor de transcripción? O sea, esto no me queda claro. También me parece hasta dónde es independiente de linfocitos T, ya sea TH1, 17, porque todo llega al día 14-16, que es el pico máximo de la manifestación clínica, y de allí incluso las intervenciones empiezan a disminuir, no las desaparecen, y ninguna hasta este momento ha evitado la magnitud de la infección. Lo que hace es disminuye un poco la enfermedad, pero no la evita.
Sí. Entonces,
son como dos partes.
Sí, entonces la primera es
sería
el trigger del UPR response, ¿cierto? Entonces, lo que vimos es, nosotros identificamos ese pathway a partir de este estudio de environmental factors, pero lo que vimos es que mismos citoquinas te pueden hacer el trigger de esta stress response en células. Y de hecho, eso es una observación común para la gente que trabaja con immune cells. Este pathway, por ejemplo, está hyperactivated en B cells que están produciendo inmunoglobulinas. ¿Por qué? Porque es un trabajo producir todas esas inmunoglobulinas. Lo mismo pasa con astrocitos. Lo que es interesante, y eso me lleva a responder tu segunda pregunta, nosotros no creemos que estos mecanismos de new immune deregulation en astrocytes pueden ser el trigger o pueden initiate the disease. En mi cabeza eso es driven by T cells, by immune deregulated adaptive responses, ¿cierto? TH1, TH17 cells you were talking about. But lo que pasa es que una vez que esas T cells o esas peripheral immune cells llegan al sistema nervioso central y trigger these inflammatory responses driven by astrocytes and microglia, estas respuestas se vuelven independientes de aquel trigger inicial. Y de hecho, eso es lo que vemos en la enfermedad. Vos podés bloquear inicialmente la patogénesis driven by T cells y B cells, if you wish, whatever, pero eventualmente tenés estas cosas que son completamente independientes. La otra cosa que eso nos sugiere es que este trigger de deregulated glial responses ocurre muy temprano durante la enfermedad, simplemente que no los ves, por un lado porque los efectos de las T-cell responses son mucho más agudos, ¿no? Tenés una lesión, tenés una pérdida de myelin, tenés dysfunction which is very acute, y la otra cosa es que también hay plasticity. Tenés múltiples mecanismos que de alguna forma te van a compensar para esa neurodegeneración progresiva. Pero eventualmente, una vez que pasas un threshold, esa neurodegeneración se va a hacer evidente e irreversible. ¿Contesté la pregunta?
Gracias.
Gracias. Bueno, igual, ¿no? Muchas gracias por la paciencia, por todas estas interrupciones. Y me sumo a lo que dijo el doctor Vianney, impresionante plática y trabajo. Dos preguntas. Una está relacionada a esa interacción y comunicación que se da entre la microglia y los astrocitos. Y específicamente a nivel de estos dos proteínas de membrana, efrina y...
Sí, FP3, FRB3.
¿Esa interacción se modifica en relación al tipo de polarización en la que se puede encontrar el macrófago, digamos, en la variedad que hay de M1, M2 y los subtipos de M2? Y supongo que habrá algo muy similar por lo que contaste a nivel del astrocito, es decir, el estado de polarización modifica esa interacción y cuál sería la repercusión en el sentido de la inflamación.
Exacto, eso es muy bueno. Si te acordás la forma en que nosotros identificamos esta interacción es enfocándonos en astrocitos los más proinflamatorios, ¿cierto? Entonces lo que vemos es que está asociado con subsets específicos de astrocitos proinflamatorios. En resting astrocitos no ves este pathway. Es solo cooptado o misactivated cuando estos astrocitos proinflamatorios son activados. Y desde el punto de vista de la microglia o el monocito, porque algunos monocitos pueden también activar este pathway, es lo mismo. Es un specific subset de microglia que lo activa. De hecho, en paralelo, analizamos otro pathway que hoy no mostré para simplificar, que involucra semaforin 4D y plexin B1, B2. Y esos son también usados por otros subsets de microglia y astrocitos. Entonces, la forma en que creo que tenemos que pensarlo es que a medida que identificamos estos subsets, inducidos por distintas cascadas proinflamatorias o whatever, vamos a encontrar distintos mecanismos de comunicación. Y creo que eso va a ser útil desde el punto de vista de tener estrategias terapéuticas bien específicas.
Y la otra pregunta, que no es necesariamente de los datos que nos presentaste y que por desviación, porque yo trabajo con el HR, quisiera hacerte. Tú publicaste un trabajo en el cual demostraste que la activación del HR promovía la diferenciación hacia células T reguladoras y que de esa manera se podía disminuir todo este fenotipo de esclerosis múltiple. Pero eso fue utilizando TCDD, esta dioxina que es tóxica. Cuando probaste FIX, que es otro agonista, que como el TCDD induce genes como el CYP1A1 a través del HR, pues la respuesta fue contraria. Entonces, como dos agonistas que están actuando con el mismo receptor ya tienen una idea de cómo eso, esas diferencias simplemente por utilizar dos estructuras químicas diferentes, pero que actúan sobre una misma vía de señalización, pueden llevar hacia supresión del sistema inmune o una sobreactivación?
Mañana voy a hablar de eso.
Perdón.
Sin embargo, sí, de hecho trabajamos obviamente con el AHR, de nuevo, yo no lo cloné, no quiero tener ese honor que no es mío, pero sí vimos que hay efectos que son ligando específicos y creemos que esos efectos están asociados con con la vida media del ligando y las proteínas que lo degradan, y por otro lado con la capacidad de ligandos de inducir interacciones con distintos factores de transcripción between AHR. Y eso creo que es central en cuanto a la habilidad de AHR para ser reclutado, no solo a AHR binding sites, pero también ser regulado, por ejemplo, STAT3 binding sites, que son muy importantes para T-cell differentiation.
Gracias.
¿Alguien más?
Buenos días, doctor. Antes que nada, pues comparto la opinión de creo muchas personas aquí, que es que la conferencia fue muy interesante. Mi pregunta va dirigida, bueno, también son dos. La primera es de si estas tecnologías y todo el conocimiento que nos estuvo mencionando, ¿cree que podrían también aplicarse para entender la interacción de células nerviosas del sistema nervioso, pero en este caso en sangre periférica? Para en este caso, bueno, con células de sangre periférica y para entender mejor la enfermedad o para desarrollar biomarcadores?
Sí, un programa que tenemos que creo que es muy interesante es usar este mismo, el SPIC-SIC, right? Esta habilidad para hacer forward screens de interaction molecules, bla, bla, bla. Entonces, para usarlo para identificar factores, moléculas que operan en la célula dendrítica para controlar la polarización de T-cells. Y creemos que eso es importante para identificar nuevos mecanismos asociados con la polarización de effector and regulatory T-cells. Como variación de eso es usarlo para tratar de identificar cómo el tumor o cómo tumores, en nuestro caso estamos enfocados en GBM, pueden controlar o afectar la polarización o la activación o la función efectora de células efectoras que te dan inmunidad antitumoral. Y la otra cosa que estamos haciendo es, mencioné esa técnica de Rabbit-seq. Cuando la desarrollamos, cuando logramos hacerla que funcione en humanos, una de las muestras que estamos analizando son gut samples from IBD patients. Son muestras con patients con inflammatory bowel disease, porque esas interacciones neuroinmunes, obviamente nosotros nos interesamos en el sistema nervioso central, pero muchas de ellas o distintas ocurren, por ejemplo, entre enteric glia and enteric neurons y T-cells, dendritic cells, macrophages. Y entonces tienen efectos locales en inflammatory bowel disease, o como voy a mostrar mañana, te van a controlar células T que eventualmente van a migrar al sistema nervioso central o a otros tejidos. A mí me interesa el sistema nervioso central. Entonces, eso es interesante.
Y mi segunda pregunta va dirigida también al HR precisamente, ya que pues también se ha observado que tiene como papel en la barrera hematoencefálica.
Sí.
Entonces, ¿usted cree que la microbiota intestinal y los ligandos que producen DHR estén ligados precisamente en la interacción de microglia y astrocitos? Sí,
nosotros, hay dos trabajos que publicó Rothammer en el laboratorio, uno en el 2016 en Nature Medicine y después en el 2018 en Nature, y lo que él mostró es que derivados del triptofano son metabolizados por la microbiota intestinal en compuestos que después son activados en el hígado y que pueden pasar la barrera hematoencefálica. y literalmente controlan la producción de una molécula que se llama TGF-alfa, el Tumor Growth Factor alfa en microglia, que controla la polarización de astrocitos. Esos dos fueron el primer reporte del control de astrocitos por el gut-brain barrier, por el gut-brain axis. Y por otro lado, es la primera vez que tenemos un ejemplo de cómo productos del microbioma del gut te pueden controlar el crosstalk entre células del sistema nervioso central. Y eso fue la regulación de estas, tanto del receptor como del ligando, mediando esas interacciones entre microglia y astrocitos, era mediado por AHR sensando esos compuestos microbianos y directamente transactivando o interfiriendo con la expresión de estos receptores o ligandos.
Okay. Muchas gracias, doctor.
Buenas tardes, doctor. Muchas gracias por su plática. Mi pregunta es acerca de la heterogeneidad que menciona sobre los astrocitos, ¿no? Sabemos que aun cuando son proinflamatorios, de todos modos, es una población muy heterogénea. Me causa duda cómo hicieron para seleccionar los astrocitos que fueran más proinflamatorios y si llegaban a haber diferencias entre las proteínas que se expresaban en la membrana que comunican con la microglia.
Bueno, entonces la forma en que lo hicimos fue, nosotros hace años reportamos el análisis de astrocitos por single cell data de pacientes, de 28 pacientes con 28 controles y 20 pacientes con MS. Y estas son muestras de distintas áreas del sistema nervioso central, de lesiones, de normally preordained white matter, so on and so forth. Y entonces lo que pudimos hacer fue identificar poblaciones que por su patrón transcripcional eran proinflamatorias, y después fuimos en ratones, we CRISPR out whatever, control them, y mostramos que no eran proinflamatorias, ¿cierto? Entonces, que eran proinflamatorias. Entonces, eso nos permitió armar lo que nosotros decimos es un transcriptional signature de proinflamatorio astrocytes, que es básicamente NF-kappa B signaling, la producción de algún tipo de citoquinas o quimoquinas o factores que llevan a la regulación de la mitocondria, y entonces tenés mayor producción de empty rows y so on and so forth. Eso es lo que usamos nosotros para pescar en nuestro single cell dataset los astrocitos que tenían el mayor factor proinflamatorio. Y lo que puedes hacer es un método bioinformático donde decís, te doy un ejemplo simple, si tenés 10 genes, si una célula tiene match para los 10 genes, tiene un peso de 1. Si una célula tiene 5 genes, tiene un peso de 0.5, ¿no? Entonces, de esa forma como pudimos rankear todos los astrocitos en nuestro dataset y pescar los que son más proinflamatorios. Una vez que hicimos eso, lo que hicimos fue buscar barcodes que estuvieran compartidos entre estos astrocitos y microglia, porque eso quiere decir que son astrocitos que están interaccionando con estas microglia en particular. Y una vez que hicimos eso, bueno, nos enfocamos en identificar moléculas de interacción. Así es como salió FB3, FRB3, Semaphore 40, Plexin B1, B2.
Gracias.
¿Alguna otra pregunta? Una más, por favor.
En relación al factor, al inicio de tu plática nos hablabas de un factor de transcripción, el XBP1,
Yeah.
Y que su sobreexpresión era importante para promover al astrocito más inmunoreactivo, ¿cierto? Más
Proinflamatorio.
Este factor de transcripción, simplemente por elevarse los niveles de transcripción, ¿es suficiente para que promueva este fenotipo o requiere de algunas otras modificaciones posttraduccionales? ¿Se puede inactivar o activar? ¿Cómo está ahí, no, además de presente, si se mueve en un estado activo o inactivo?
Entonces, eso es muy bueno. Este es un factor de transcripción que se regula por splicing alternativo. Entonces, uno puede regular la expresión de la transcripción del gen, y en condiciones normales, o si no hay activación del pathway, el gen se va a transcribir, el RNA va a incluir un intrón, y ese intrón tiene un stop codon. Entonces, y eso está al principio del RNA. ¿Por qué es importante entonces? Porque cuando el ribosoma empieza a producir esa proteína, inmediatamente se encuentra el stop codon y genera una proteína que es corta, but useless. ¿Okay? Entonces, cuando el pathway se activa, por ejemplo, por este sigma-1 signaling, lo que ocurre es hay un evento de splicing alternativo. este intrón is spliced out, y entonces el ribosoma, cuando está leyendo ese RNA, puede ir all the way y entonces codificar, generar una proteína que es full length y es functional. Así es como se regula la activación, no solo a nivel de RNA, pero a nivel post-translational. Now, la otra cosa que es interesante es que nosotros vimos que XBP1 generalmente interacciona y potencia el recruitment de NF-κB to target genes.
Muy bien, muchas gracias por su presencia y los invitamos a que vengan mañana también a las 12. Y muchas gracias, doctor. Una estupenda charla.